Hello, everyone. My name is Angel Flowers, and my OUR project was the population distribution analysis of tardigrades found on Quercus virginiana, or the southern live oak. Tardigrades are ubiquitous microscopic eight-legged lobopods from the clade Panarthropoda. They are closely related to arthropods, oncophores, and nematodes. They thrive in moist microhabitats such as moss, detritus, and lichen, requiring only a thin film of water to be active. At the introduction of water, tardigrades will rehydrate and continue their life cycle from where they last left off. When conditions are no longer favorable, they can again return to the ton state, where they slow down their metabolic activity, essentially pausing life for years at a time if necessary. Tardigrades can enter one of two latent or ton states. The first is cryptobiosis, and there are four types: anoxybiosis, cryobiosis, osmobiosis, and anhydrobiosis. The second state is diapause, which leads to encystment and resting eggs. These dormant states allow for them to be carried on the wind and in water droplets on the wings and feet of migratory birds. Here in Texas, we get those birds such as hummingbirds, Baltimore orioles, and several species of swallow that migrate through yearly. Sabine Woods Bird Sanctuary was chosen for sampling because it's stopover for migrant birds between the United States and southern areas across the Gulf. Because this position is also barraged with warm, moist southerly air most of the year, these factors were believed to increase tardigrade density and diversity in this area. It also happens to be home to a Cheniere ridge populated by coastal southern live oaks. So 5 of the largest, oldest oak trees on the property were chosen for sampling. We sampled the moss, lichen, and detritus across each of 4 tree features: limbs, trunks, crotches, and roots. This was done by scraping or scooping the sample from the substrate into a paper bag. Sampling sites were chosen randomly, and directionality, as well as height from the ground and distance from tree base, were recorded. We got the samples back to the lab, and to process them, we took 1 gram of dry weight sample and placed it in a 2-ounce condiment cup with 20 milliliters of water. These were allowed to soak for approximately 24 hours, in which we placed 3 1-ml drops into a separate dish and observed them under a microscope in order to remove all the tardigrades and place them on slides for viewing under microscope. Excess tardigrades that were removed from the sample were set aside for DNA. So, single factor ANOVA demonstrated that 4 tree features drive significant variation in both relative density and species richness. 2-factor ANOVA was run to test the importance of individual trees, features, and their interaction on each parameter, relative diversity, and species richness. Neither trees nor their interaction with features explain significant variation for either relative density or species richness, which means that we had good replicates in our trees, which is exactly what we wanted. Sample relative density positively correlated with species richness, meaning mean relative density was not significantly different among most features types, except branches had significantly greater mean density than the roots, and mean species richness in the branches was significantly greater than in the crotches, but there was no significant difference between other tree features. Out of 60 moss samples, 321 tardigrades and 6 eggs were found, encompassing 4 genera and 6 species of the superfamily Macrobiotidae. Mesobiotis harmsworthy made up 55%, making up the largest percentage of tardigrades found. Minibiotis intermedius made up 36%, and together this was 91% of tardigrades found in all samples with just 2 species. Of note was Macrobiotis evelinae. This is a species that was last described by Dept in Brazil in 1938. It was listed in the 2021 edition of Actual Checklist of Tardigrada as nomen incorrendum. So this would be the first discovery of Macrobiotis evelinae in North America and Texas. Okay, and then here are some of the variation. I just wanted to show everybody what all different species look like, and how closely they looked, and how the differences are. You can notice a few small differences in the buccal apparatus and the macroplacoids. I do have a couple eggs on here, G and D. These are two different species with egg. And so A was Macrobiotis echinogenitus. This is Minibiotis intermedius on B. C was Macrobiotis evelinae, and D is the egg of Macrobiotis evelinae. E, F, and G are all Mesobiotis harmsworthy species. This is a little bit of variation in the buccal tube. One's a little bit wider and shorter than the other. And then you have the egg, which has a lot of ornamentation compared to D. H is Paramacrobiotis tonali, as well as I. K is also Paramacrobiotis tonali, and it's right here behind my head. I don't know it, but these are just, again, variations of the same species. So it's very difficult to identify tardigrades simply for the fact that there is minor variations between species and within species. So you have to like notate all these specific little details to try to really pinpoint which species you're dealing with. And then, of course, J is Paramacrobiotis areolatus. That is it. I would like to give a special thank you to the OUR program for the funding for this project. Special thanks to the Texas Ornithological Society. They gave me permission to sample out at Sabine Woods, and they were very gracious with that. Also, big thank you to Alyssa Broussard for all her help with the sampling and for sorting and just with everything. You were an excellent help. Thank you so much. I would also like to say thank you to my mentors, Dr. Miller and Dr. Hoch. Dr. Hoch provided so much practical advice and was on point whenever I needed him. Dr. Miller was a big help with morphological identification of all the different species. Also, I would like to thank my family. Without their support, I would not have been able to do any of this. So a big thank you to my husband and my kids for all the emotional, mental, and physical support at home. Thank you so much, and I hope you enjoy my project, and I hope this interests you in tardigrades. They are very easy to work with and a lot of fun, and there's so much room for discovery. Thank you.